Hello, dear students. Today we will be discussing about the fourth step of recombinant DNA technology. In the previous lecture, we mentioned about how we do the isolation of the DNA from a cultured cell, and then how we just separated or the desired gene or DNA fragment with the help of restriction into nucleus. And in the another step, we just inserted that desired gene into a vector, that's a plasmid, to make a recombinant DNA. Now, in the fourth step, let's try to insert this recombinant DNA into a host cell. In this step, we just take the recombinant DNA or the desired DNA fragment or the recombinant plasmid. If we just can do this with the help of two methods, either we can do it directly or indirectly. Indirectly, we just introduce this desired DNA fragment into a vector, that's a vehicle DNA or a carrier DNA, and we just introduce or insert this desired gene or desired fragment into that vector, which is actually that desired gene, which is also called a passenger DNA, to make a recombinant DNA. This is indirect method where we use a vector. So that's called a vector mediated transfer. We can also introduce the desired gene or the fragment of DNA directly into the cell or transfect into the cell, mostly in the eukaryotic cells of animal cells, with the help of many methods which we have already discussed, like there is direct gene transfer, that is gene gun transfer, or biolistic or micro injection and all that. But here most commonly we use the vector mediated transfer, that's plasmid vector. So what we actually do here is that the plasmid free bacterial cells, we take the bacterial cells, Escherichia coli, we just remove the plasmid from these cells, and then we made a recombinant plasmid, recombinant DNA, and then these plasmid free cells, bacterial cells, they are made competency, their ability to take up the foreign DNA, their competency is enhanced by certain methods which we already have discussed, like electroporation is done there, or treatment of the cell wall with calcium dipositive ions. So all these methods we make the cell competent, and these plasmids having the recombinant DNA and the selectable marker, in case of plasmid, for example, ampicillin resistant genes, they are introduced into the host cells, which is formally called a transformation. So transformation of these plasmids are done as the plasmid enter the bacterial cell. The host cell becomes ampicillin resistant. Now we get those bacterial cells with these which are transformed, they definitely have a resistance gene with that's ampicillin resistance. They will show resistance towards ampicillin. So in order to differentiate or separate transformed cells from non-transformed cells, we just grow them in ampicillin medium. Ampicillin is actually an antibiotic, and we know that antibiotics work against the bacteria, they just kill the bacteria. And when we grow these bacterial colonies inside in the ampicillin medium, all those bacterial cells will die which are not transformed. That means we do not have taken up the recombinant plasmid with that resistant ampicillin resistant gene which acts as a marker. So in this way we can get separate the transformed from non-transformed cells. So these transformed cells or recombinant DNA based cells are used in bioreactors. Now we are going to just transfer these transformed cells in bioreactor. So the fifth step which we are going to, that's the we have to obtain the foreign gene product. Ultimately what we are mean for is to get the uh, you know the desired gene product, because the final product of a gene is a protein. So the major aim of recombinant DNA technology is to produce a desired biochemical or protein useful to humans. It is actually, what is a bioreactor? It's a container in which the biochemical process is carried out by using living cells, and their growth medium is called bioreactor or fermenter. Actually, a bioreactor is generally 100 to 1000 liter cylindrical metal container with a covered base to facilitate mixing of contents. So in a bioreactor, we just put all these contents, the transformed bacterial cells. We place the growth medium there. We place certain salts and vitamins and hormones and all those things which are needed for the successful growth of these bacterial transformed bacterial genetically modified bacteria, which we have performed in the previous steps. And finally we are doing all these things in a bioreactor, simply an instrument, a cylindrical instrument used to get culture all these cells. Finally, when we just transform these, when we just put these transformed set in a bioreactor, we get the desired product. What we are going to do now finally is the downstream processing. So what actually is the downstream processing? It's actually the recovery of the product from the fully grown genetically modified cells. It means that once you know bacteria have multiplied, they have replicated, they have produced, you know the replicated forms, division of the cells have done, and the genes have copied itself, and finally we will get that the product from that desired gene. Then we have to do its purification. That product is purified and preserved. This product is separated, purified, and then formulated with suitable preservatives in order to preserve it. And after preservation, after separation, purification, it is we go we are, it has gone through clinical trials are then performed on this product to know its use, whether it is it has this efficacy. Can it be used for the mass introduction to the people? And first of all, it is, yes, it is use is checked, its efficacy is checked by doing the trials in the laboratory on animals and then on humans, and finally it's put into the vials or bottles and just sold to the companies and other worldwide to the people which are affected. For example, if we are just making an insulin product from an insulin gene, this is how recombinant DNA technology is meant for producing a desired product in this manner.